Thank you, everyone, for joining this month's Muscle Health Research Center seminar. We're very happy to have Dr. Jeffrey Dilworth with us. And before I introduce Dr. Dilworth, I'd just like to go over some quick reminders. When you have questions, if you could please save them to the end of the presentation. You're welcome to raise your hand. And I'll cue you to ask your question verbally. You can turn your camera on or not, it's up to you. Alternatively, if you could please post your questions into the chat group directly, publicly, rather than to Dr. Dilworth. On that note, it's an absolute honor and privilege to introduce Dr. Dilworth from Cellular Molecular Biology Graduate Program, the University of Wisconsin in Madison. Dr. Dilworth was previously at the Ottawa OHRI, the Ottawa Hospital Research Institute, since 2004, and has recently moved to the University of Wisconsin. Dr. Dilworth has been extremely prolific in the field of epigenetic regulation of gene expression during mammalian development and regeneration, making huge impact in multiple high-profile journals. And we're very, very happy to have him here today and to learn about the epigenetic adaptation of satellite cells to the injured muscle environment. So on that note, Dr. Dilworth, if you wouldn't mind taking over here, you have, thank you very much for joining us today.
Thank you very much for having me. It's a pleasure to be sort of virtually back at York. It's been a few years since I've been there, but definitely a fun place for me to visit, and good to see some colleagues' names on the screen here.
Wonderful. And your screen's looking perfect. We can see it.
Okay. I'm just going to reset up. You can see my laser pointer?
Perfect.
Okay, great. Well, thank you very much for the introduction. It's a pleasure to be here. I'm going to start off today by showing this slide, which is really to remind everyone that in the response to a muscle injury, you're going to have a lot of different cell types carry out the regeneration. So this is a freeze injury that had taken place, but you can see very quickly after a few hours, you've got necrosis of the muscle fibers happening, and then an infiltration of other cell types. And the muscle stem cells themselves are not going to carry out regeneration until after 2 days of regeneration. And in the meantime, you've got all these other cell types coming in that are coordinating the regenerative process. And so I want you to keep that in mind as we move forward through this discussion today. Now, we used to think of the satellite cell as being this really small round cell that didn't have very much cytoplasm associated with it. It was mostly nuclear. But our vision of the satellite cell has changed over the last few years. This work here out of Rob Krause's group has really shown that in the quiescent state, the satellite cells have these long protrusions, and it's also shown down here. You can see that it sort of wraps itself around the satellite cell, and it appears as though it's sensing its environment to know when the injury has taken place so it can activate itself. Now, my laboratory is interested in epigenetics, and the satellite cell, of course, is living over here on the left in the quiescent state most of its life. But in response to an injury, over the first 2 days, it's going to become activated. And approximately 40 hours after injury, it's going to enter into cell cycle. Those cells are going to proliferate. When you have enough cells to carry out the formation of new muscle fibers, you're going to get terminal differentiation. These cells are going to fuse together, and you get the multinucleated differentiated myotubes over here. Now, each one of these cells along the pathway from the quiescent state over to the differentiated state all have the same DNA inside of them, but each state's going to have a different set of genes which are being expressed. And our concept going into establishing my lab 20 years ago was that epigenetics would be involved in controlling the cell fate transitions between the quiescent and activated state, the activated to proliferative state and the proliferative to the differentiating state. And so our goal over these years has been to try and identify what are the epigenetic enzymes which would control the ability of the satellite cells to move one way or the other in this process. And if we could identify those, would there be the possibility of being able to modify the ability of these cells to transition in either aging or disease? Okay. So I talked about the many different cell types that are present in the regeneration. You've got your quiescent satellite cell up here at the top. It's a slide borrowed from Tom Rando. And you can see not only do you have the satellite cell moving through myoblast to the myofibers, but you've got macrophage being present, fibroadipogenic progenitor cells that Fabio Rossi identified. You've got the endothelial cells, parasites, Tregs. All of these different cell types are releasing cytokines, growth factors, many different signaling molecules, which are going to determine the timing of these satellite cells moving through the proliferative stage and the differentiating stage. Now, I'm going to tell you two stories today that we published recently. One of them is talking about really the epigenetics, but this first section that I'm going to talk about is talking about a process that maybe not all of you know about. It's called promoter proximal pausing, and it's a transcriptional regulatory mechanism that is thought to allow the genes to activate very quickly in response to different stimuli coming from the extracellular environment. And just conceptually from the literature, the fact that this polymerase would be stalled on the promoter of developmentally regulated genes led us to believe that it might be important in the process of muscle regeneration. So what exactly is promoter proximal pausing? Well, we always talk about the ability of transcription factors to recruit onto promoters the transcriptional machinery required to activate gene expression. And this would be the general transcription factors here, as well as the polymerase. But what happens at a lot of developmentally regulated genes is that approximately 20 to 60 base pairs downstream of the transcription start site, you get a stalling of the polymerase, and the polymerase is sitting there and waiting for signals telling it to move into the gene. And while the polymerase is sitting there, it's actually oscillating back and forth. And of course, you've got the RNA tail sticking out the backside of that polymerase. And as it's oscillating back and forth, that tail becomes susceptible to nucleases that can then cut off these first few base pairs of the transcript. And if you get that happening, it's going to be an abortive transcription event that occurs. So proteins such as NELF here are going to be important in stabilizing these transcripts that are associated with the stalled polymerase. And that's what we're going to look at today. What's the role for this NELF protein in regulating the ability of the satellite cells to move through from the quiescent state through to the differentiated state? Now, as I said, NELF is this negative elongation factor and is made up of several different subunits: NELF A, NELF C, NELF B, NELF E. And if this is the polymerase crystal structure here, what you can see is that NELF really comes and gives it a big hug down at the bottom side of the molecule here. This is the stalled version of the polymerase as it's been elucidated in crystal structure. And over here is the elongating form, where the NELF complex gets replaced by the PAF complex, which is an elongating factor. So the NELF here would cause steric hindrance for the PAF being able to bind, but we also know that NELF is going to protect the RNA that is sticking out the backside of the polymerase and prevent it from being degraded by the exonucleases. So we came into this with a very simple question: does NELF-mediated stabilization of the nascent transcripts play a role in regulating muscle satellite cell function. And for this, we went and we turned to a model that many people use in the field, which is the PAX7 Cree-ER mixed with the Cree LOXP recombination system. We got this allele from Karen Adelman at Harvard. And what happens specifically in satellite cells is you get the expression of this Cree-ER. And when you add tamoxifen, the Cree-ER is going to make its way into the nucleus, and it's going to bind to these LOXP sites, and eventually they will delete the exons which are present in between those flock sites, and you're going to remove the NELF gene. And we use this system here where TD tomato, which had a stop codon in front of the transcription start site, that stops codon gets removed. So cells that underwent recombination are going to begin to express TD tomato. And you're going to need that information for looking at some of the slides, how we know where the satellite cells are. So the first question we asked, what happens in response to a cardiotoxin injury if you don't have NELF? So here we have the NELF knockout. And when I show you this picture, I guess I'm hiding the labeling here, but this is the control contralateral leg, and this is the leg that had received the cardiotoxin treatment, and we've waited 28 days after. So the muscle is pretty much fully regenerated after 28 days. Looking at this, it looks like fairly healthy muscle, but it's only when you compare it to the wild type that you see that there's a problem. So the contralateral leg looks the same between the wild type and the knockout, but you can see here in the injured leg 28 days later, these muscle fibers are quite a bit smaller than here in the wild type leg. That's quantitated over here. But where you really see the difference is when you look at the whole muscle. Here's the cardiotoxin treated wild type muscle, and here's the cardiotoxin treated NELF-B muscle. The muscle looks fully regenerated. It just came out very small compared to its wild type counterpart with the weight shown over here on the right. Now, what's the reason why those fibers are so small? We went and we isolated single fibers and we stained with DAPI, which is going to show the DNA. We can see where the nuclei are. And you can see that in wild type, you've got these centrally located nuclei, which are very tightly packed along the wild type myofiber. You can see that the spacing is much further apart here in the NELF knockout. And what that means is you have less nuclei within each muscle fiber, therefore less transcripts are going to be made, less protein being made. And so that hypertrophy of the fiber is due to the fact that there's a lot less protein present within those cells. Okay, so we saw that regeneration was occurring, but occurring not to the same extent. Less cells are fusing into the myofibers. But what happens to the satellite cell compartment of those muscles that had been repaired? We went and we looked both 7 days and 28 days after injury, and our cells that are satellite cells are stained here with PAX7 in red, and you can see them up here in the wild type state. But there's very few of them down here in the NELF knockout, and that's quantitated over here. So approximately 10 per field of view here in the wild type conditions, and we're down to about 2 per field of view in the knockout conditions. So we've gone and we've regenerated muscle. We've got a few satellite cells, but not very many left. What's going to happen if we go and carry out a repeat injury? And what you see happen here is that we gave cardiotoxin at day 0, cardiotoxin again at 28 days, and we sacrificed the mice at 56 days and looked at the muscle. Here with the H&E staining, you can see very good regeneration in the wild type, but very, very poor regeneration occurring just because of the fact that we've depleted the stem cell population within that NELF knockout mouse. And yeah, just a really ugly looking muscle, lots of interstitial cells, fibrosis happening within that muscle. So that tells us that you don't want to lose NELF. Okay. So one of the nice things about muscle regeneration and why we really like working with this system is that we can move ex vivo and start to look at the stages of regeneration and find out where the problem is. So is it in the activation of the satellite cells? Is there a problem with proliferation, or do those cells fail to differentiate? Now, we didn't think that there was going to be a failure of differentiation, only because I take you back to this figure from before. We saw really nice regeneration occurring. We were just getting smaller fibers. So it didn't occur to us that differentiation might be a problem. And in fact, if we put NELF knockout satellite cells in culture and look at their differentiation, they form multinucleated myofibers without any problem. So trying to assess where in the regenerative process there might be a problem, we looked at the activation of the satellite cells. So the activation looks at the entry of the cells into cell cycle. And as I said before, this happens approximately 40 hours after injury. So what we can do is cause a cardiotoxin injury in the mouse, and we can inject EDU. EDU is an analog of deoxyribonucleotide, and as the cells start to replicate the DNA, that deoxynucleotide analog is going to get incorporated into the DNA, and then we can use click-it chemistry to add a fluorophore onto that, and we can look at it by FACS. We can see which cells have entered into replication and therefore cell cycle. So if we wait 28 hours after the injury, and then we do a pulse of EDU for 12 hours, we can see which cells have entered into cell cycle. And as we look at the FACS analysis here, looking at cells that are both TD tomato, so the satellite cells, and EDU positive, you get an equivalent amount of about 50% of the cells have entered into cell cycle. So that's not where the problem is. We wanted to try a similar approach to look at proliferation of the satellite cells. And so here we waited until the cells had actually entered into cell cycle. By 60 hours, they're rapidly replicating, and replications occurring about every 8 hours from 40 hours onwards, up to 4 days. So by waiting to 60 hours and then doing a 12-hour EDU pulse, we can get a sense of how much proliferation is going on in these cells. And when we look at the FACS analysis here, you can see that 50% of the wild type cells are incorporating EDU, but that drops down to about 37% over here in the NELF knockout. So that's telling us that there's a defect in the expansion of these cells. Now, this experiment here was done in vivo. We can visualize this ex vivo as well by plating out muscle progenitor cells and looking at EDU incorporation. You can see that here. And we can just count the number of cells that are growing in culture each day. And you can see that the NELF cells are not expanding very well compared to the wild type. So it really is the inability of the satellite cells to expand their numbers that is hindering the size of the myofibers, which we're able to generate from the NELF knockout. We did do some single cell RNA-seq analysis, and this is more confirmatory because we already realized from the straightforward experiments that there is a proliferation defect. But here at 72 hours after injury, so the cells should already be in cell cycle by 72 hours, we went and carried out single cell RNA-seq analysis. And this here is a combined analysis of both the wild type and the NELF knockout. We were able to do a pseudotime trajectory, and we can see that they start out here at the quiescent satellite cell stage, move up into this hyperproliferative area at the top of the arrow, and then come down to myogenin positive cells that are about to differentiate down here at the bottom. And what you can see in the wild type conditions at 72 hours, almost all of the satellite cells are accumulated up here in the hyperproliferative clusters, shown up here or up here. But in the NELF knockout, you can see that the cells have started to move out of that hyperproliferative stage and appear to be going through to the differentiation stage, and they're undergoing a premature differentiation, which is leading to those smaller myofibers. So how does NELF regulate the proliferation of the muscle cells? We went and we did different types of genomic analysis. Here on this slide, I'm looking at ProSeq, and what ProSeq is going to analyze is the nascent transcripts that occur. So if we're doing an RNA-seq experiment, what you are getting is a snapshot. You're getting the transcription that's happening at any given time, but also any transcripts that are stable that are going to have a lot more molecules than transcripts that are less stable. But here, if you use ProSeq, you can actually measure the transcript as it's being made by the incorporation of these biotinylated nucleotides. And then we can isolate using biotin and sequence this in order to see how different genes, their expression is being affected. And so when we did this ProSeq in collaboration with Karen Adelman at Harvard, what we found was that, in fact, in the absence of NELF, you did have a little bit of movement of the polymerase and therefore transcription moving into the gene, so a little bit more here, but that wasn't statistically significant. Where we did see statistical significance was in the genes that were downregulated. You can see that in the wild type, you have a lot more transcript up here at the transcription start site where the stall is, and you have a lot less transcript here in the NELF knockout. And what that means is that in the absence of NELF, you're losing the stability of many of these transcripts. And we're getting this abortive transcription that I talked about in the beginning. So we did do RNA-seq in addition to this ProSeq and looked at genome-wide binding of NELF using a procedure called cut and tag, as well as looking at the polymerase using some chip sequencing. And we went looking for genes that showed changes in the ProSeq signal, so less of the nascent transcript, but also less accumulation in the RNA-seq, and that there would be NELF binding at that site where the positive polymerase would sit, as well as polymerase and reduced amount of polymerase in the absence of the knockout. And so one of the genes that we identified is PEDF. We identified quite a few genes. You can see here that genes that are upregulated are involved in extracellular matrix, MAP kinase signaling, TGF-beta signaling, whereas genes that are downregulated have more to do with ribonuclear complexes, which might suggest protein synthesis, but also P53 signaling down here and cell cycle. And these are expected based on the fact that we had a proliferation defect. So this is published, so I'm not going to go through all the experiments that we did for this, but what we found basically were that two main target genes that were downregulated in the absence of NELF were PEDF that I just showed you, but another one here called cyclin G1. And in normal cells, when you have both NELF, when you have NELF in the normal cells, you're going to produce PEDF. You're also going to produce cyclin G1. Cyclin G1 is a partner in binding with this phosphatase, PPA2, and it's going to sequester the PPA2. But if you don't have NELF, PEDF is not signaling into the P53 signaling pathway anymore. And also you don't have the cyclin G1. In the absence of the cyclin G1, the PPA2 is going to dephosphorylate MDM2. And in the absence of MDM2, P53 tends to accumulate because MDM2 is a ubiquitin ligase, which is going to continue associating with the P53, ubiquitinolate it, and cause it to turn over. So in the absence of this turnover, because cyclin G1 is not present, P53 is going to accumulate. You're going to get transcription of genes such as P21 here. And P21 is a cell cycle inhibitor. and it's going to prevent the cells from cycling and cause them to undergo differentiation. Okay. So what we found from this study was that NELF is necessary for allowing the expansion of these muscle progenitors here, and that in the absence of NELF, these cells are going to undergo premature differentiation. But we also saw that NELF may play a role in the repopulation of the niche, because when we knocked out NELF and we looked at the regenerated muscle, there was a lot less satellite cells being present associated with that newly regenerated fiber. And this led us to ask the question, well, what role would NELF play in the repopulation of the satellite cell niche? And so one possibility is the fact that NELF-B might just directly regulate the ability of the satellite cell to return to the quiescent state. Now, this wasn't something that we really thought was going to be the mechanism, just for the fact that we didn't see any defects at all in the activation of the satellite cell. We only saw defects when we were looking at the proliferation of the satellite cell. So, but of course, we can't rule that out. But we started thinking about it, and another possibility is that NELF actually isn't necessary for this repopulation of the quiescent state, but it had more to do with this inability of the satellite cells and muscle progenitors to proliferate and have enough cells to carry out the regeneration. There might not be enough cells left to move back, and so that this would be an additional step in the process of movement back to the quiescent state. And by this, what I mean is that in normal wild type conditions, you might say that this one satellite cell proliferates enough times that we make up an arbitrary number of 10,000 cells is necessary to carry out good regeneration, and that 9,950 of those would go into the making of the myofiber, and stochastically 50 of those cells would move back into the quiescent state. And perhaps in the NELF knockout, because you have this proliferation defect, you're not getting 10,000 cells, you're only getting about 400 cells. If we look proportionally, that means that 398 of them are going to fuse into fibers. We're going to get these fibers that are less, have smaller diameter associated with them, smaller caliper, and it would also reduce the number of satellite cells that were able to repopulate the niche. To address this question, we worked with Michael Rudnicki's group at the OHRI, and we did some transplantation experiments. And for these experiments, what we did, we took immune-compromised NSG mice and we irradiated them to kill off as many of the satellite cells as we could in these mice. We then took either wild type or NELF knockout satellite cells from our TD tomato positive mice. We took 12,000 cells and we injected them into an NSG mouse that had not received any injury. So we depleted their satellite cells with irradiation, but there's no injury here. We waited 21 days after we did this transplantation, and then we started serial cross sections through the TA and started looking for satellite cells. And the first thing you'll notice in these pictures is that the number of fibers which are staining red is much larger here in the wild type compared to the NELF knockout. And this is to be expected because of the fact that the PAX7 positive cells can proliferate more in the wild type condition compared to the NELF. And so the number of TD tomato positive cells shown here is less. But when we went looking for satellite cells in the satellite cell compartment, we did in fact find lots of satellite cells that were associated with the red fibers, both in the knockout and in the wild type. And if we went and we corrected the number of PAX7 positive cells to the number of TD tomato fibers, what we saw is that there was no statistically significant difference between the number of satellite cells which had repopulated the niche in either the wild type or the NELF knockout. And so what that tells us is that the satellite cells can repopulate the niche if they're not required to move into the muscle fiber for the regeneration. And so this suggests that in the case of a large muscle injury, the satellite cell itself is not going to be egotistical. It's not going to say, I need to regenerate myself right away, which is the case that does happen if it's just a cell division for a minor repair, as Dr. Rudnicki had shown. What happens when there's a major injury is that the satellite cell says, we're going all in on this repair. We're going to allow for the satellite cells to all move into this regenerative state, continue proliferating until we have enough satellite cells in order to fuse and make myofibers. Once we have those myofibers, some of the remaining cells are going to move back and repopulate the satellite cell niche. And the results that we have with this is consistent with studies that came out of Brad Olwyn's group at the University of Colorado, where he was feeding the mice EDU in their drinking water. And he did this either at days 0 to 2 or days 4 to 7. And they then isolated the muscle fibers and looked at the number of cells that had EDU incorporated with them, meaning that they hadn't gone through any more cell cycles. And you saw almost no satellite cells which had EDU if you added the EDU between day 0 and day 2. However, day 4 to 7, when all of this proliferation is going on, that is where the cells were coming from that were EDU positive that were repopulating the niche. And so that tells us that in normal conditions, what's going to happen is that wild type cell is going to have NELF sitting on these cell cycle genes and regulating the entry of the polymerase into those genes to keep that cell cycle process running smoothly. When you do have an injury, this satellite cell is going to become activated. It's going to proliferate extensively, make enough satellite cells to actually go in and repair the muscle fiber, but also go and repopulate the niche. And because of that, you're going to get a normal, healthy myofiber. Now, in the absence of NELF, you're going to get this abortive transcription occurring, and you're going to get a lot less expression of these cell cycle genes. In that case, when your satellite cell here responds to injury, it's going to activate and start proliferation, but you're not going to make as many of these cells. It's not going to be able to incorporate as many new nuclei into those myofibers, and you're going to end up with much smaller caliber myofiber, and it's going to most likely have less strength. We haven't done that test, but certainly those mice have much smaller muscle. So there's inefficient myo repair and no repopulation of the niche if you don't have NELF. So these experiments here told us a few things about muscle stem cell biology. And one is that the satellite cells have gone and taken one of the many different transcriptional mechanisms that exist in mammals and placed it inside the satellite cell and said, we want you to regulate the ability of the satellite cells to expand their population. And it does that by regulating the stability of P53 regulated genes or associated genes. And when NELF is present, that transcriptional pausing is going to be responding to signals from the extracellular environment to decide how much transcript should come off of these genes regulating P53. Now, loss of RNA Pol2 pausing on its own is not sufficient to prevent repopulation of the niche. So even though we have satellite cells not repopulating the niche in the NELF knockout after an injury, we know from our transplantation studies that, in fact, those satellite cells can repopulate the niche. It's just that there's not enough of them to fully repopulate the muscle after the injury. And finally, that depletion of the muscle stem cell pool in the niche is caused by a loss of progenitors that have undergone premature differentiation rather than the active role for NELF in reentry of the cells into quiescence. Okay, so that's the end of the first part of the talk. I'm going to take you through another study that we published more recently, which has to do with an epigenetic control. So the main point that I want you to get from the talk that I'm giving today is that there are many different regulatory mechanisms that decide the transcriptional output from different genes within the cell. And the ability of the cell to carry out regeneration does not depend on a single process or a single mechanism. There are many different mechanisms that are going to be carrying this out. And these mechanisms are not specific to muscle cells. They're present in all cells. But the stem cell populations, as they saw a need for being able to regenerate efficiently, they've gone and they've coerced some of these different mechanisms into individual processes in order to regulate the ability of the satellite cell to regenerate. So the second part I'm going to tell you about has to do with polycommon trithorax group proteins. And I don't know, I don't see any faces, so I don't know which people know about epigenetics and which don't know about epigenetics here, but no matter how much you know about epigenetics, you probably know that H3K27 marks are repressive marks. They basically sit on top of genes and say, do not transcribe these genes. They're thought of as being a brick wall to prevent genes from being transcribed. And there's, of course, one enzyme that puts in place the H3K27 marks, EZH2, but there's two enzymes which are able to remove that mark, UTX and JMJD3. And I've actually talked at York before about our studies on UTX, where UTX is necessary for the terminal differentiation of satellite cells. But all of these different stages of muscle regeneration also express JMJD3. So that at every stage of regeneration, the satellite cells are going to express both UTX and JMJD3. And this had us wondering, do they play redundant roles, or are they doing different things in order to regulate the process? Now, the fact that we would have two different enzymes is strange, because in yeast, they have transcriptional repression, but they have no H3K27 marks. Drosophila have H3K27 marks, but they only have UTX. They don't have JMJD3. And if you have a mouse that doesn't have UTX or JMJD3, their development looks fairly normal through to day 10.5. So really, what is the role for these enzymes, and why does a muscle cell require two of them? So the first thing that we did was confirm that satellite cells, which are TD tomato positive here for PAX7, also have the H3K27 mark, and every cell that is red has JMJD3, and every cell that is red also has UTX. So both of these are present. So do we require both of them? For this, we turned back to our cardiotoxin injury model. And what we found was that compared to the control that regenerated very well, the JMJD3 knockout and the UTX knockout had a lot of interstitial cells remaining, and we had smaller myofibers. And what that tells us is that JMJD3 cannot compensate for UTX, and UTX cannot compensate for JMJD3. So again, we're going to go back and look and see which stage of regeneration you need JMJD3 for. I already told you that we published previously that UTX was necessary for the terminal differentiation of the satellite cells. So you can see that in the absence of UTX, satellite cells have poor differentiation, quantitated over here. Whereas the JMJD3 knockout satellite cells, they can undergo terminal differentiation just as well as the wild type. They look very nice, these multinucleated fibers. We went and we carried out single cell RNA-seq analysis, this time focusing in on 30 hours post-injury. And the trajectory of this single cell analysis starts down here at cluster 0 and makes its way into the proliferating cells here, which start, you can see the proliferation signatures in this upper part of the trajectory, while the activated cells are down here in the lower part of the trajectory. And what we found was that wild type satellite cells moved very quickly through the activated state into the proliferative state, and UTX knockout cells did similar. However, if we look at the JMJD3 knockout, you can see here at the earliest point in the pseudotime trajectory, you've got an accumulation of JMJD3 cells that are not present in the wild type or the UTX. And what that suggested to us was that there was a activation defect in the JMJD3 knockouts. So we came back to this activation assay that I told you about before, where we use EDU clicket chemistry and we inject the cardiotoxin, wait 28 hours, and then do an EDU injection for 12 hours and look at the incorporation of EDU into newly synthesized DNA. You can see here in the wild type state, we've got about 50% of the cells that entered cell cycle. It's not significantly different in the UTX knockout, but we do have a very significant decrease in the ability of the JMJD3 knockout cells to enter into activation, and that's shown down here. JMJD3 works in activation, not terminal differentiation. UTX works in terminal differentiation. Now, does JMJD3 act through its ability to remove the H3K27 mark? We actually have a mouse which has 2 point mutations in it, in the Jumanji C domain, which is the domain which is required for the removal of methylation marks. And you can see that the protein that is demethylase dead is produced, and its levels are not decreased compared to the wild type. So if we use those mice and do the activation assay in exactly the same way, you can see that just these 2 point mutations are sufficient to prevent the cells from entering into cell cycle efficiently. So that tells us that the demethylase activity of JMJD3 is required for regeneration. And the real reason why we ask this question is this Jumanji C domain is only about 10 kilodaltons, whereas the protein of JMJD3 is 160 kilodalton. It has both enzymatic and non-enzymatic functions, but this experiment here confirmed to us that JMJD3 requires its enzymatic function in order to carry out regeneration. Now, this is where the project became interesting because Kieran in my lab had to do the experiment probably about 15 times before we finally figured out what the solution was. But of course, we wanted to understand the mechanism. And so we took FDB fibers, which are fibers out of the foot of the mouse. We took those fibers out, put them in culture, and we looked at the activation in the absence of the rest of the mouse. And we could see that wild type and JMJD3 knockout, or wild type and demethylase dead satellite cells on these fibers were activating just as well as the wild type, and that had us perplexed. And of course, that's why Karen did the experiment 15 times, but 15 times it gave us the same thing. We then thought about some experiments that had been done by Tom Rando's group a few years ago, where he had described some cells called G-alert. And what G-alert is that you cause an injury in the one leg, and satellite cells in the other leg become, they don't enter into cell cycle, but they become alerted to the fact that they might be needed in the future. And so they become, they move a little bit out of quiescence. So that told us that that there was communication going on between the satellite cells. So what we decided to do was to cause a cardiotoxin injury in the one leg of the mouse, wait 36 hours, and then isolate muscle fibers out of the other leg of the injured mouse. So these are fibers not from the injured leg, but from a mouse that had previously been injured, and whatever signals were causing that alerting might make their way over. And what we could see was that the wild type activated very well, but unlike in the uninjured state, all of a sudden we were seeing a defect in the ability of the JMJD3 knockout, as well as the JMJD3 demethylase dead to activate if we'd caused a previous injury in the mouse. What that tells us is that there was a cellular response to the stress of systemic inflammation. There's inflammation signals coming from this one injury, and it's affecting the other leg over here. We could reproduce this with an extract from damaged muscle. So we could isolate the satellite cells from an uninjured mouse associated with these explanted fibers. And as I showed you up here, there was no ability of the wild type to perform better than the JMJD3 knockouts. But if we made an extract by grinding up an injured muscle 30 hours after injury, spun out all of the cellular debris and just took the water-soluble extract there and included it in the incubation, all of a sudden these satellite cells from uninjured muscle had a difficulty entering into cell cycle if JMJD3 was not present. Okay, and so how does JMJD3 allow the muscle satellite cells to integrate the signals from the other cell types within the niche? We went and we carried out RNA-seq analysis, and what we were able to see was that JMJD3 regulates approximately 315 genes, and we also saw that it was involved in the upregulation of a lot of genes. And these are probably indirect targets, because if you're not removing H3K27 marks, you should get downregulation of genes, not upregulation. But when we look at these indirect genes, what we saw was that there were a lot of genes involved in inflammatory response and chemotaxis, innate immune response. And what that suggested was that there was a cytokine storm going on within that muscle that did not have JMJD3. And what we were able to see was that there was an increased infiltration of CD45 positive hematopoietic cells into the injured muscle, as stained here in green. But we were really interested in the direct targets of JMJD3, which of course are the blue ones over here on the left. And we wanted direct targets that had JMJD3 bound. So we used a technique called cut and tag to look at genes that had H3K27 marks, as well as JMJD3 being present. So knowing that genes that would be important for this process were not regulated by UTX, but were regulated by JMJD3, we looked at the RNA-seq for genes downregulated in the absence of JMJD3 that would accumulate H3K27 marks in the absence of JMJD3, but also had JMJD3 binding to the promoter region. And we liked this gene here called HAS2. HAS2 is a gene that makes hyaluronic acid. And so you get the polymerization of N-acetylglucosamine and glucuronic acid, just a polymer of one after the other. And high molecular weight hyaluronic acid has anti-inflammatory effects. And so this led us to follow this more closely as a potential mechanism for how JMJD3 was allowing this communication. And this just shows that hyaluronic acid synthesis is not happening in the quiescent satellite cells, only in the activated satellite cells. And hyaluronic acid is actually degraded by the counteracting enzyme called HYL1 in quiescence, and that gene there goes away as you activate the satellite cells. And here what we found was that the satellite cells, shown in red, are encoded in the encapsulated in this HABP hyaluronic acid in wild type conditions, but the satellite cells do not have hyaluronic acid around them in the JMJD3 knockout. And this is just a cross-section showing the same thing. 30 hours after injury, you've got the red satellite cell with yellow around it because of the hyaluronic acid. You don't have that yellow ring around the satellite cells here in the JMJD3 knockout. And we can look at the we can look at the ability of hyaluronic acid to be involved in the regenerative process by blocking its synthesis. And we can do that with a small molecule called 4-MU. This 4-MU blocks the synthesis by being a competitive inhibitor for the UDP GLC-UA here. And so if we were to inject the 4-MU into a regenerating muscle, you can see that we've got very poor regeneration. And if we look at the activation of those satellite cells, that's the part of the regenerative process that is being affected by the loss of hyaluronic acid synthesis. But interestingly, if we block hyaluronic acid synthesis in satellite cells, if we just take those satellite cells from an uninjured muscle and we don't have any muscle extract associated with it, 4-MU does not block the activation of the satellite cells. But if we have the cytokines, the inflammatory molecules from that damaged muscle, and we block hyaluronic acid synthesis, all of a sudden we do have this block in activation. And most importantly, what we were able to show is that this activation defect in the absence of JMJD3, if we carry out the reaction in the presence of exogenous hyaluronic acid, we can rescue the ability of those satellite cells to undergo activation. I'm just looking at my clock and I'm should be finishing up, but I'm just going to say that we used our RNA-seq and we found that interferon gamma was the signal that was coming from the injured muscle, because if we added the damaged muscle extract or interferon gamma to the activation of the satellite cells in the absence of JMJD3, we were seeing an inability of those cells to activate. And so what we think is happening is that early on in the regenerative process, we've got these inflammatory cytokines that are inhibiting the ability of the satellite cells to enter into cell cycle. And we're exploring right now if this might be a mechanism contributing to the timing where the satellite cells don't actually enter into cell cycle until 40 hours after injury. But eventually what's going to happen is that about 20 hours after injury, you start making hyaluronic acid synthase, which is going to make the hyaluronic acid, which is going to incorporate into the extracellular matrix. And that can associate with receptors and either increase or decrease binding affinity. And we think that in this case, it's decreasing the ability of interferon gamma to signal into the nucleus and block activation, and therefore allows the cells to enter into cell cycle. And so I'm just going to end with this slide here. We found previously that UTX removes H3K27 marks put in place by EZH2 to prevent the transition to the differentiation state. In the absence of JMJD3, we've got poor movement of the cells from the activated to the proliferating state, and that is due to the fact that we've got all of these different signaling molecules coming from the macrophage that are present there. Okay. So what this has told us is that hyaluronic acid provides the muscle satellite cells with an extracellular matrix that is wired to protect the satellite cells from the hostile environment associated with the regenerative niche, meaning that there's a lot of cytokines and reactive oxygen species that are really preventing the satellite cells from activating, and by surrounding the cell with hyaluronic acid, now they can activate. And I think more surprising to us was the fact that satellite cells, being a rare stem cell population and a small percentage of the total number of mononuclear cells present in the regenerative niche at 30 hours after injury. The fact that those cells can regulate the magnitude of the inflammatory response was very surprising to us. But if you don't have JMJD3 in those satellite cells, you get twice as much inflammation occurring within the muscle, a cytokine storm going on. And so that suggests that the satellite cells are very important for communicating with the rest of the niche. So with that, I'm going to thank Kieran, who did the work with the JMJD3 knockout. I see that I put a new slide up here and left Dan Robinson's name off of here, but he did the NELF story that we talked about today. And of course, the lab has moved to Wisconsin, and people interested in muscle who want to come enjoy a winter which is similar to that in Canada, you're always welcome to come join us in Madison. So with that, thank you very much. I'm happy to take questions.
Thank you very, very much, Jeff. Excellent presentation. Tons of data. I have so many translational questions that I think would just be demonstrating that what you've done is set a foundation for physiologists to just explore ad nauseum and different diseases. So I won't go there. Rather, this is for the audience. So everyone here, thank you for being here. If you are interested in asking a question, please raise your hand, and I can unmute you. You're welcome to show your video. You don't have to. Alternatively, you're welcome to put the question in the chat. So while you're thinking of questions, I'm happy to ask my own. This last point, actually, this isn't a question I had really thought through, so pardon me if I'm still working it through, Jeff, but
Yep.
this last summary slide with the hyaluronic acid sort of shielding the satellite cells. You know, immediately I thought of the only thing I know about HA, which is knee therapy, osteoarthritis, which no expert, but I understand it's more of a lubricant and shock absorber, for lack of a better term. But I've heard it's an anti-inflammatory for the knee. So not that there are satellite cells in the knee joint, but this concept of shielding satellite cells from immune cells. Do you think that's a ubiquitous phenomena throughout the body, or?
Yes, I think that is going to be the case. A collaborator of ours, some new work that we're doing where we're specifically expressing the hyaluronic acid synthase in satellite cells. This collaborator who made the mouse has actually looked at making hyaluronic acid in every cell, just ubiquitously making additional hyaluronic acid. And what they found was that in the gut, there's less leaky gut because the satellite cells are able to repair the intestine a little bit better. They found increased longevity. This was a Nature paper that was just published about a month ago with these mice. And they didn't get as much into mechanism, but they found increased lifespan, increased health span. Many different tissues looked a lot younger, a lot healthier, probably because of the fact that the satellites or the different stem cell populations were protected from inflammation by the hyaluronic acid. So
is it possible that it's actually acting, for lack of a better word, reverse to the immune cells, macrophages and so on, to block their secretion of cytokines? Or are the cytokines still being secreted and somehow it's blocking the activation of the cells, or inhibition, pardon me?
Yeah. So it's a very good question, and I couldn't give you a definite answer on that.
Yeah.
But what I would, my thinking right now is that it's probably protecting the satellite cell from the signals that are coming from the individual different immune cells, whether it be neutrophils or macrophage. And the reason that we think that is that we have tried injecting hyaluronic acid into the regenerating muscle the same way that you talked about injecting the hyaluronic acid into the cartilage there to try and protect. And we did not see any beneficial effects of that. And what we think is happening is the mechanism which hyaluronic acid uses for synthesizing, that HAS2 uses for making the hyaluronic acid, is kind of an inchworm type ratcheting. It just keeps pushing and pushing a little bit more out each time it adds another polymer onto it, and it sort of weaves its way through the collagens and the fibronectins that are in the extracellular matrix. And work that's coming out of cancer, hyaluronic acid is involved in the metastasis of cells. What they found there is that things like c-Met receptor binding with HGF, it will form a quaternary structure with CD44, which is a receptor for hyaluronic acid, plus the hyaluronic acid, and the four of them being together stabilizes things and allows for a much stronger signal coming from there. And we think that in that way, it can stimulate things like HGF signaling, but in the same way it could block IGF signaling is kind of the model we're working with, but it's guessing right now.
That's very interesting. I'd like to look more into this HA and muscle regeneration. I saw a hand go up first from Anthony Shime. Anthony, go right ahead.
Hi, Anthony.
Hey, hi, Jeff. How are you?
I'm great. Looking forward to talking with you after.
Yeah. The NALF-B is its gene expression in quiescent stem cells? Do you find it in quiescent stem cells?
Yes. Yes. So it's expressed everywhere ubiquitously, and it sits on almost every gene in the genome. And yeah, I think that what we thought when we went into the experiments, we thought that NALF was going to play a role in the actual activation step, that as soon as there was an injury, there was going to be a signal that said NALF, go away, and let a lot of transcription happen off of these genes. We were surprised that that didn't happen. But yes, it's present in all cells. When we did the cut and tag analysis, we saw that it was bound to about 6,000 different genes in the muscle cells, but only about 100 of those genes changed their expression when you knocked out NALF. And so what we're trying to figure out now is what are the signals that are coming from the extracellular space that are being integrated. NALF, not the B subunit that we knocked out, but NALF-E has about 30 different phosphorylation sites on it for different kinases. And so it allows for a multiplicity of different events occurring to different signals is how we're thinking about it.
I was going to ask you what the upstream signals were that were regulating it. Yeah. Okay. I guess you answered my question regarding, because I wanted to know if there were different subunits, but you're saying it's on every gene. So it's really specific because there has to be a turn on signal and then a turn off signal.
Correct. Yes. So
one particular gene.
Yes. Yeah. So one of the turn off signals is phosphorylation by a molecule called PTEF, which is a 2-protein complex, one of them being the kinase. And so you could have the turning off on specific genes because PTEF interacts, say, with MyoD, but it interacts with a lot of different transcription factors. So if MyoD were to bring PTEF into this gene here, it phosphorylates the NALF, but also the polymerase. NALF goes away there, and it's just a small number of the 6,000 that get turned on.
Right.
So that would be the mechanism, one of the mechanisms for limiting the number of genes. And in the same way, it could be PAX7 does it in different conditions, maybe beta-catenin in other conditions. So.
All right. Thanks, Jeff.
Welcome.
Okay. John?
Hi, Jeff. Thanks for such a great talk. Really incredible science you've done in the last few years. Just wanted to ask you about a little bit about the JM-JD3 story, going back to that. It is quite surprising, that only one target gene, the hyaluronic acid, is responsible for the whole knockout of JM-JD3. So there are a couple of things related to that. What about the rest of the genes that are demethylated by JM-JD3? Are they playing a role in satellite cells? And probably the more interesting question for me is what's recruiting JM-JD3 to its target genes? Have you looked into that?
Yeah, both very good questions. I'll start with the recruitment. The recruitment, we looked underneath the peaks of the JM-JD3 and found that we looked at sequences that were there, and you can't find any A's or T's. It's almost all G's and C's, which rules out almost every transcription factor, because almost every one of them has at least one A in there. We think that it's recruited to CPG islands based on that result, but we have to prove it. It's just an observation. When we looked at the motifs for the 50 most highly represented motifs, there wasn't a single A in any of them. So I'll answer that there. The other part of the question was, what are all the other genes doing? Yes. So I've told you that hyaluronic acid is the most important gene ever. We had lots of different newspapers asking us if the popular press asking whether all these skin creams that have hyaluronic acid were going to make their muscles better. But we actually don't think that hyaluronic acid does everything. So hyaluronic acid, we think, is playing the role of protecting the cells from the cytokine signals that are preventing the activation from occurring. We now have the Has2 knockout mouse, and we're able to reproduce that with the Has2 knockout mouse. They have an activation defect, but the full knockout of Has2 actually has additional defects that we didn't see in the JM-JD3, because of course we just had reduced expression, not complete loss. But what you don't see in the Has2 knockout is the cytokine storm and the increased infiltration of the immune cells. So other targets are responsible for that. And so we've got some new knockout mice that we just saw that we were able to generate yesterday that we're going to, it's a gene that's involved in regulating the amount of reactive oxygen species in the extracellular space around the satellite cells. It's really highly expressed in satellite cells and downregulated in the absence of JM-JD3. So we're thinking that it's playing a role probably in the cytokine storm that's occurring. That there were only about 50 genes that showed altered gene expression only in JM-JD3, but also the H3K27 marks accumulating and had JM-JD3 as a direct target. So you can actually, it's a usable number that you can go through the literature and see what each one does and try to imagine how it might contribute to the phenotype. And so we've kind of been doing that, but we've got 3 or 4 that we think are most representative of the phenotype there, and we're working on those.
Right. Yeah, if there are no other questions, I will follow up with one, because I'm sure that, we've got a lot of people here who are interested in muscle function and exercise and so on. And I'm sure Chris was wanting to ask this question, but held on to it. But the idea then that that these mechanisms that you've proposed here might not just be operative in regeneration when you have serious muscle damage, but what about, if let's say in an exercise model, do you think that these mechanisms will be at play? I mean, I'm not quite sure where the current consensus is on how much satellite cells contribute to exercise responses, but maybe you can just give us your take on all that, if you have one.
It's another great question, because of course we've caused a major injury in the muscle with the cardiotoxin. How often is the hyaluronic acid going to be necessary? We have done some preliminary experiments with Benedict Chazot's group in Lyon. It wasn't really an exercise model itself, but using electrical stimulation of fibers and going with higher energy rates to cause more and more damage to them. There we could see the satellite cells. It's a smaller activation defect, but we could see what was trending towards a significant effect, but we didn't quite hit significance yet with the number of mice that we've gone through yet. Of course, there is interferon gamma, which accumulates in the muscle after some strenuous exercise. So we think that it probably is going to play a role there, but it's going to require some new experiments. We do, since we moved to Madison, one of the other muscle researchers here is Troy Hornberger, who does a lot of exercise physiology, and so we're going to get the JMJD3 mice over to him and see if he can help us solve that problem.
Yeah, great. Thank you very much.
That's a perfect set of hands, Troy Hornberger, for that idea.
Yeah, he's a great guy. I like interacting with him.
Yeah. Thank you. If you don't mind, Jeff, we've got a student question that's messaged to me directly, and it's a two-part. And some of the things I was wondering as well, and then we'll certainly let you go. I was also very interested in the translational aspect of NELF. Is there any disorder or disease known to be associated with NELF?
Well, no, NELF itself, if you had a complete inactivation, would be lethal.
Okay.
So I don't think that there's ever been anything described. It doesn't mean that it doesn't exist, right, but nobody has pointed to it as the cause yet.
Well, another example where sort of foundational knowledge to begin to explore the degree to which it's being activated too much or not activated enough, as opposed to being absent in various disorders. Yeah. And
in the case of muscle, if the person has muscle and they're just smaller, you might not think that much about it. Right. But it has different effects in every cell type, so it does become lethal. If it were a germline mutation, you wouldn't see a germline mutation come through of NELF, but if you had a somatic mutation happen, you might not notice it. So
okay. Well, that comment might segue a little bit to the last part of this question, which was, I wonder how this protein may impact other aspects of muscle aside from satellite cell activation, such as normal global transcription under altered phenotypes and conditions and so on.
Yeah, another great question. I'm going to go back to a little bit to the answer that I gave to Anthony before, in that we know that it's only a small number of the NELF target genes that are changing their gene expression when you don't have NELF during the activation or the proliferation of those cells there. But that's an integration of a single signal coming in. And the NELF-E has about 30 or 40 different phosphorylation sites on there. We think that in different scenarios, different signaling molecules are going to cause phosphorylation events that are going to regulate the NELF activity. So you might find that in the muscle fibers that we haven't looked at yet, in the fibers, maybe the response to during a hypertrophic response, maybe NELF is important for turning on a certain set of genes, or just any time that you're changing the extracellular environment, you're going to activate new signaling pathways that have to be integrated into the genome and the changing of genes that are being expressed. We know that in the inflammation case, it's going through JMJD3. In whatever growth factor is stimulating proliferation, it's going in through NELF. And just the cell has chosen different regulatory modules within the transcriptional machinery for different processes. And I'm sure that NELF is going to be involved in others. We think that NELF might also be important for sensing changes in metabolic profiles of the cells as well. We're right now looking at the role of 3 different AMPK phosphorylation sites on the NELF-E. Just because when we did our RNA-seq, a lot of the target genes are also metabolism genes.
Wow. Okay. Metabolic reprogramming, regeneration, now a NELF link to that.
Yes.
Oh, that's excellent. That's exciting. Of course, I study metabolism, so of course I'm excited. All right.
We can talk about it another time offline.
I would love to do that. Yeah, that's right. Everybody, please join me in thanking Jeff, Dr. Dilworth, for being here today and teaching us all this wonderful stuff, because I think we need to keep talking to you.
Thank you for your time. Thank you for the great talk.
Oh, thank you. Have a great day, everyone. Bye.
Thank you. And if you could just stay on, Jeff, and
Yeah,
everyone else.
I'll be with Anthony.
Yeah, that's right. Everyone else, presentation is now over. Thank you very much for joining. Have a lovely weekend.
See you, Jeff. Nice to see you again.
Bye, John. Bye, everyone.